Well, why don't we get started? I'm sure other people will be joining, and I have to keep an eye open for the participant list to see if there's anybody in the waiting room. But we got a good number of people on the line already, over a dozen, and I'm sure there will be more joining over time. Welcome to all the new people, and of course, all returning faculty, friends, students, postdocs, whatever. Welcome to the fall semester of our Biostat series. As if you've been following the email announcements, the schedule is already set up through early October, but I would really welcome additional contributions. Just drop me an email anytime, dthomas@USC, if you would like to volunteer. What we're going to do today is kick off the fall with the first of two seminars describing our large program project grant on statistical genetics. So we'll go through sort of the first half of that today. Second half will continue next week. And then we'll move on to other topics. But our goal here was to try to introduce the faculty and their interests so that you can get an idea as to who to, who you might want to approach for mentoring and future research. So let me explain, let me first share my screen here. Whoops. Let me admit somebody else first. Here, is there, while I'm sharing my screen, I will not be able to see the participant list. Is there somebody else that can monitor the participants and admit anybody that's coming late?
I can help with that, Duncan.
Thank you, George. Okay, so
you can co-host, don't you, to do that?
Yeah, you may need to make one of us co-hosts, Duncan.
Yeah, I better do that right away.
George is co-host.
Let's see.
In fact, you'll need to make a couple of us co-hosts anyway to be able to show slides later.
Right. So I'm doing you right now. That's okay. Here we Paul, you are now co-host. And where's Huaiyu? Well, I'll deal with Huaiyu later. I can't seem to get to the participant list now that I'm in this mode. So let me explain what we're going to do today. We are in the process, we are now in year 5 of this 5.5-year project and are starting to work on the submission of our competing continuation proposal, the renewal, which we will be submitting at the end of next month. So because this is what NIH calls a program project grant, meaning a collection of 3 or more related RO1 size projects linked around a common theme, it's a big process submitting one of these grants. There is the so-called ARA letter, which is a request for permission to submit a large grant, which we have now submitted and are waiting for formal acceptance of that permission. But prior to that, there is a conference call with a variety of NIH staff at which we present our ideas and discuss with them. And on the basis of that discussion, we may have modified some of our plans. So what we're going to do is go through some of the slides that we used for that NIH presentation. And the focus will be on our ideas for the future rather than very much about our accomplishments in the first cycle. Of course, we will be following that up with various full-length seminars as the fall proceeds, where we describe some of the work that we have been doing. But there won't be very much of that today. The focus will be on our ideas for the future. I think that was all I really wanted to say by way of setting the stage. Jim, can you think of anything else I should have said before we get started?
Yeah, I think that covers it.
Okay, so the project is we have called IMAGE, and that stands for Integrative Methods of Analysis for Genomic Epidemiology. And as I say, we're speaking about the renewal now. So what we're going to try to do today is just give you an overview of the theme of the project, briefly walk you through what the current project consists of, that we will come back and talk about in more detail in future seminars, and then describe our thoughts about the renewal. I think that's enough for now. Why can't I advance? There we go. So our overall theme is to develop novel statistical methods for this so-called post-GWAS era, where we're now faced with a flood of new omics types of data. And our idea is to try to integrate this information across different data types. So that means, on the one hand, across different types of omics on the same individuals. So in one study, we might have genomics, we may have exposome information, metabolomics, expression, and a variety of different data sets, all of which are high dimensional. And we want to look at the relationships amongst those different omics. We also want to look across phenotypes. Our focus here is particularly on cancer, but we're well aware that various omics are related to multiple cancer types, and there may be other phenotypes like, let's say, obesity, that could be related to cancer as well. And finally, across species, so we want to use information that's available on various experimental platforms to inform the analysis of epidemiologic data. And another area that I think is somewhat unique to our work is that our goal is to incorporate the sort of wealth of information that's available out there on the web in terms of what is already known about the biological functional variants or experimental information that we might be able to bring to bear on the analysis. Ultimately, we want our methods to be useful. So by that, we mean able to improve the detection of novel risk factors for cancer, develop risk prediction models using high dimensional data, and genetically targeted interventions, screening, treatment, and so on. So here's the sort of picture that summarizes overall what we are doing. As I say, our goal here is integration. And in the first cycle, we had 4 projects, because it's still going on. Project 1, which has been led by David Conti and Juan Pablo Lewinger, deals with hierarchical models for integrating genomic and functional data. Project 2, led by Paul Marjoram and Paul Thomas, uses phylogenetic models to try to infer gene function from available experimental information that's stored in databases like the Gene Ontology. Project 3, which I've been leading with the help of Josh Milstein, looks at the internal environment as a mediator of genetics and external exposures, and develops techniques like Bayesian network analysis to try to accomplish that. And finally, Project 4, which has been led by Kim Sigmund and also Paul Marjoram, uses these same kinds of phylogenetic ideas, but to try to model the growth of tumors and their genetic evolution over time. These projects, 4 projects, are supported by 4 cores. One of them is an obligatory administrative core, and then it's supplemented with a core led by Huaiyu describing the various data resources that are available for annotating genes, variants, proteins, etc. There is a core C having to do with high dimensional data analysis, computationally intensive methods, and trying to develop some standards for programming and so on. And that's been led by Paul Marjoram. And Core D, that's been led by Jim Gauderman, that organizes the various data sets as well as the software and makes them available to the general public. I think enough said about this picture. So there again is the list of the 4 projects that I just went through and the 4 cores. I can't think of anything more to say about them right now. There's been quite a lot of productivity from the first cycle. At least that's the message that we want to convey to NIH. Here's just a sampling of a few of the publications that have emerged from that work. I won't talk about these now, but if you stay tuned to future talks, I'm sure you will be hearing from various of us describing this work in more detail. So just some more publications. We have also been very productive in terms of software development, and these have been published in journals like Bioinformatics and made available on our own GitHub repository, as well as CRAN and various other sources.
Are you on it, Glenn?
Mm-hmm.
Okay, so what I want to do now is move towards from what we've been doing to the future. So again, these are the 4 projects, and each of these, and we have reorganized them into 4 new projects, which are loosely based on the past, but with a number of different contributions. So this doesn't describe all of the links, but our new project, which is going to be led by Juan Pablo, is heavily involved, based on the work of Project 1 from before, but it also includes links from several of the other projects and relies as well as on some cores. Project 2 will be led by David Conti and now focuses more on mediation. Project 3 it will be led by Jim Gauderman, and it focuses on gene-environment interactions. And you'll be hearing more about that from him just momentarily. And finally, the new Project 4 combines elements of the old projects 2 and 4 and a few other things. Whoops, sorry. But with a new focus on gene characterization, involving work on gene function, tumor heterogeneity, and a new aim on novel methods for gene set enrichment. So with that, I'm going to now turn it over to Jim. I will continue to advance the slides as you want, so just let me know.
Okay. All right. Thanks, Duncan. So this is a revised schematic, very similar to the last one in form. You just heard about the 4 projects there. And a couple of the big changes here, we had focused on colorectal cancer as the primary application in the last round, and we've since gotten involved in more research, and more research has evolved to have the kinds of data that are interesting for this project. So we're now expanding to be more generally to other cancers. And then cores B, C, and D in the lower right are going to be taking really have as a focus getting out general use programs and software tools so that we can get the work that we do out there for others to use and make sure it's not just staying in the archives of some methodological journal. All right, next slide, please. Oh yeah, so if we want to try to boil down what each project is doing, the first project is really focused on how we can estimate parameters and make predictions using multi-omic data, and we'll develop a lot of the core methods that'll be used by other projects. P2, next slide, is going to be focusing on how multi-omic data can be used to disentangle gene outcome relationships, so kind of getting into the nuts and bolts of mediation and kind of pathways of how genes affect the outcome. Next slide. P3 is going to bring in environmental data and focus on gene-environment interactions. And then P4 will kind of focus on pathway modeling, both for individuals and also branch off of work we've done this first round on tumor modeling. And I mentioned that we're going to broaden the scope for cancer. So we have a number of data resources that are available to us. There's the multi-ethnic cohort, which several of you may have heard of in the past, and you'll hear about plenty of that through courses and other research seminars. This is an ongoing cohort that's been around for a couple decades, and it's got a strong focus on cancer, obviously, and a lot of evolving genomic and other kinds of omic data. So it'll be a real goldmine, I think, for use now and in the future. There's several clinical trials that are being conducted. So I won't say too much about those, but a lot of interesting data, including some GWAS data and other kinds of omics data as well. Next slide. There's the Orion Network, which is a nationwide network of kind of clinical data and a variety of additional data that are available. Next. The Respond cohort is a cohort of 10,000 African-American men that Chris Hyman and David Conti are leading, looking at a whole list of variables you can see listed there, and a lot of GWAS and other omic kind of data available. And then there's the Fiji data, which was mentioned earlier. So this is an ongoing case control study that a number of us are involved in of colorectal cancer, about 130,000 total subjects with GWAS data, lots of well-harmonized environmental data on the full cohorts, and expression data from a couple sub-studies that we're interested in using to inform a variety of these analyses. Next slide. Next slide. So Core B, for example, the annotation core, has developed a really nice web tool that they call AnnoQ, which can reach out and grab data from a number of sources out there, manage that, and you can give it, for example, a range of SNPs on a particular chromosome, and then go out and access a variety of data fields for those SNPs and produce the results in a nice table that can be downloaded and used for downstream analyses, summarized, et cetera. So it's an R package, and it's going to continue to be the tool that we use going forward to make sure it's up to date with the latest and greatest data, and that it's user friendly and becomes a nice tool for people to use. Core C is the computation core, which has really done a great job at kind of the best practices for putting together software. So unit testing, documentation, lightweight code, portable code, fast code, et cetera. They're the kind of place to go to kind of make sure that you go along with all of those best practices in putting software together. Next. And then Core D has been involved in the past in software deployment and data acquisition. We're kind of re-envisioning this more now as a research translation core. So it's going to have a more prominent role in educational activities, supporting seminars, still getting software out there, perhaps using Shiny apps and R Markdown to make software a little bit more approachable by outside groups. and that kind of focus for this core. Next. There's an admin core. We just make sure everything happens. So that's, we can go to the next slide. Next slide, this is kind of details. I want to point out that, you know, we've been at this for a little over 4 years now, and this is the list of trainees that have been involved in the project. So you can see it's a very long list. We've had both postdocs and research associates, RAs, working on projects, cores, doing dissertation work, doing postdoc work, getting publications, making presentations. It's really been a nice place for trainees. So I encourage, you know, new students to get involved, and feel free to approach any of us if you're interested, and we can tell you more about the various activities and see whether it might be a good fit for you down the line in terms of your future research plans. And we have a good time. So we have an annual retreat. This is us at the retreat, and we, you know, talk science and have nice dinner and enjoy each other's company. So, although couldn't do that this year for obvious reasons, unfortunately. Okay, and then this was our pitch to NCI, that by funding a big, huge program project, that they were going to get more than if they funded these things separately. So I won't belabor that. That's more of a pitch to them. Okay, where did we want to go next, Duncan?
I think you were going to talk about Project 3. Do you have your own slides up, or do you want me to show them?
No, I can do it. I'll do it here.
Okay, so I've stopped sharing.
Okay, so can everybody see that?
Yep.
Okay, so Project 3 is the project on gene-environment interaction. Here's the formal title. I'll be leading it, and my co-investigators are Juan Pablo, Meredith Franklin, and Duncan. Oh, I guess I can put this in display mode here. That ought to make it better. So it is going to use centrally this Fiji data set, which I mentioned a little bit earlier, is a big data set, case control, and has a lot of well-known environmental risk factors for colon cancer that have been harmonized and brought together from over 50 contributing studies. A very interesting part of that, of this study, is the aim to try to bring in gene expression data and help use that to identify novel G by E interactions in genome-wide scan mode. So there's public data available through GTEx, and then there are 2 experiments that are kind of done within the context of this study. One involves about 400 normal colon biopsies and SNPs expression exposure data. And so it allows us then to build models for expression as a function of SNPs, environmental factors, interactions, and to then try to identify SNPs that might exhibit gene-environment interaction on expression. And those might be good candidates then down the line for SNPs that could interact with exposures to affect colon cancer. And then there's a very interesting study that Graham Casey, who's now at the University of Virginia, is doing, where they take what are called colon crypt cells or organoids from the lining of the colon. They can grow those up in essentially a petri dish kind of format and make copies of them. So you can have paired samples that are genetically identical, and they're using 35 matched pairs, lines of this from 35 different individuals, and then dosing one organoid from each pair, say with aspirin or alcohol or what have you, and leaving the other one unchallenged. And so you can look at differential expression in this very controlled environment to see which exposures might turn on a gene or enhance gene expression. And again, those might be candidates then for downstream gene environment interaction work. Okay, next slide.
That's you.
Oh, I got used to that. So we're going to develop 2-step approaches for discovering interactions. This keys off of some of our past work, but now we want to try and bring in omics data. Juan Pablo is going to lead an aim on kind of bringing this all together in a joint hierarchical modeling that can incorporate functional and experimental data. And then we're going to make sure that we have time set aside to apply these to real data and maybe find something novel. So this is kind of the standard thought about how we might draw a gene-environment interaction, and it's nothing more than a particular gene and a particular environmental factor that together have a synergistic effect on some outcome Y. And we can write the basic model as we've all seen in either a linear or logistic regression or even a survival model format. We've developed a number of approaches in the past that I don't really have time to go through that try to enhance the standard test of beta G by E that you would get out of a linear or logistic regression to basically prioritize SNPs using additional information. And again, I don't have time to go into that, but it provides a paradigm then for thinking about how we could bring in yet additional sources of information that might be brought to bear. And so things like the organoid study or the gene expression sub-studies are the kind of thing that we're thinking about bringing in test statistics that are derived from those other resources and using those to help prioritize our testing for G by E in a case control data set. We have a couple software programs that John Morrison has produced, a binary dosage program that's on CRAN and a G by E scanning program that's soon to be on CRAN that can be used to efficiently manage and analyze in genome-wide fashion and do all these tests that I've kind of highlighted in large sample sizes. So here's a revised picture of the one I just showed. And the basic idea is now we have additional data, call it X, which could be gene expression or metabolomics or what have you. And so the diagram then gets a little bit more complicated where we can still have G by E direct interactive effects on the outcome, but we can also examine G by E effects on expression or expression by E effects on the outcome. And so the goal of this project, aims 1 and 2, is to take this kind of a picture and figure out in a statistical methods sort of way how to capitalize on those relationships, ultimately to find new genes that interact with environmental factors. AIM 2 is going to use some methods that Juan Pablo has developed in the current project 1 of the current cycle, which essentially uses lasso type penalties to manage the dimensionality of the data when you're trying to bring in outside functional data like gene expression or what have you with genome-wide SNP data. You can imagine the matrices get quite large, so you need a way to kind of manage that and reduce the scope. So using lasso or elastic net kinds of approaches he's developed, and with one of his prior students, Garrett Weaver, who just graduated last year, developed a very nice package called XRNet, which implements the basic approach. And he's going to be then capitalizing on that going forward and expanding it for G by E analysis. And so the basic idea then is to add penalties in here, which will penalize essentially the regression coefficients, which are multidimensional, by additional information coming in through gene expression and the interactions there. So that's the Z matrix. The exposures are of interest, and in the G by E literature in general, there's a lot of attention paid to G and not very much attention paid to E, with the exception of a couple papers out there. So, E can get quite complicated. It's actually harder to measure than G in most situations. And so we've been thinking a little bit about how to make risk scores out of multiple environmental factors that may be correlated with each other. Things like, something as simple as principal component or a more supervised approach that can give us a poly-environment kind of risk score. And then Meredith has done a lot of work thinking about GIS and other approaches to integrate over space and time. So here we're trying to use satellite data or ground-based measurements to build exposure surfaces, and that can be used to enhance what we can typically get in more rudimentary form out of questionnaires in some of these studies. So we're going to try to publish a lot. We're going to develop software and hopefully have some nice applications back to this Fiji data set and maybe other data sets as well. And we're going to work with the other projects and the cores to get this all done. And that's the great thing about a program project grant, is that we have this synergism and common thinking across projects and cores that allows us really to do more as a group than what we could do separately. So I think I'll stop there, and I will turn it over to Paul, I think, to talk about P4.
All right, thank you very much. Just get organized. Okay. So yeah, I'm leading Project 4, which is about a variety of methods for characterizing the genome. Somehow I got put in charge, but the real work is going to be done by the people you see on the screen and people who are working for them. Oh, hang on a second. So the 10,000 foot view is that there are 3 aims, talking about different types of characterization essentially. The first aim focuses on automatic annotation of gene function, and in particular injecting a bit more biological reality into those methods than currently exist. The second aim is about pathway analyses, which I'll define in a second, and making them a bit more robust. And then the third aim focuses on epigenetic variation, or in particular regions in which there is no variation, in regions in which the epigenome is conserved. So here's my attempt to explain specifically aim 1. So this complicated figure is showing various things. The tree on the left is showing how different copies of a gene, be they in different species or sometimes multiple copies within the same species, are related to each other evolutionarily. So hopefully you can see my mouse, but if you pick a couple of leaves, so each leaf here is a copy of the gene, that are close to each other, then that's telling you those 2 leaves or those 2 copies of the gene are very closely related. Whereas if I pick 2 that are very far apart, so one down here and one at the top, say, that's telling you there's a lot of evolutionary time between them. There's an army of industrious scientists who are doing experiments in labs trying to tell us whether a given gene does or does not have a given function. And so that's the little bar on the right-hand side where you see red and blue lines. Each line corresponds to a leaf of the tree, so a copy of the gene in a particular species. And whether that bar is red, blue, or empty, essentially, is indicating whether or not there has been experimental evidence that that gene does, if it's blue, or does not, if it's red, have that function. And lack of evidence is just what's left, the white stuff. That picture is a little bit misleading in the sense that annotation is actually very, very sparse, much less common than is indicated in this particular example. So there's an awful lot of copies of genes in species that we just don't know yet, or at least we have no experimental evidence for what they do. So what we're trying to do is develop a statistical method to allow us to fill in those gaps, essentially. And so that's indicated by this gray region on the right-hand side. And the intuition is, if you're in a region where all of the genes that you have evidence for have been shown to have function, then you would guess that, or more formally, we would infer that closely related gene copies would also have that function. And that's what this picture is trying to represent. This is our estimated probability of having function, and blue is indicating we estimate it to being very likely to have function. In regions in which there's a bit more red, we're estimating the other way, that they also have no function. In regions in which there's no annotation, we essentially know nothing. So that's indicated by the white thing that's covering the middle line, which is 0.5 probability of having function, essentially. And then also in regions in which the annotations are a bit more mixed, you see less certainty. So we're inferring probably red here, but not sure, than you do in regions in which you see just one color, for example, this blue region. So the goal is to develop a complicated mathematical statistical model to describe the way genes evolve to allow us to produce those inferences on the right. And I'm going to give you no details of that model at all because of the time available. But just to say that there's a pressing need for such a model because there's a sparsity of actual experimental annotation. Existing models, including our own, holding our hands up, make all sorts of simplistic assumptions just to make everything work in finite time. We're about breaking some of those assumptions and still making things work in this cycle. In particular, most of those models assume independence either between different offspring genes from a given parent species or across different genes. We know that's not true. It's just an assumption we make for convenience. But given that it's not true, we'd like to inject that reality. So that's what we're going to try and do this time. So there are 2 parts of this specific aim, essentially about breaking 2 of those independence assumptions. Either the independence assumption across multiple genes so that we can infer them jointly, so borrowing strength, which is always a good idea when actual data is sparse. But also recognizing that different copies of a gene don't evolve independently, particularly when there's multiple copies within a given individual. What will be the product of this? We hope massively, well, we know, massively increased annotation, albeit probabilistic annotation, using a biologically principled model. Our model is because, and the hard work here, I should point out, has been done very much by George Vega-Yon, and he has invested a lot of time making the code here run fast enough that we can afford to inject some of this reality without having everything grind to a halt. And also the framework, because it's a formal statistical framework that we're building, will allow us to actually do tests to answer questions like, are these genes evolving independently? So specific aim 2, here's my complicated figure to try and explain what that is. This is about something called a pathway analysis, or a specific instance of that is called a gene set enrichment analysis. You've done some experiment, maybe you've done a gene expression experiment, and you've come up with a list of genes that are interesting. Perhaps have changed expression between cases and controls, for example. But you have a lot of genes and they have complicated names and you're trying to make sense of it. So one thing you can do is recognize that genes don't act in isolation. They actually act in complicated networks or pathways in which they interact with each other. So a question you can ask to make sense of such a gene list is, if I go through the known pathways one by one, do I find pathways that have more genes appearing in my list than I would expect by chance? And this top half of the figure is representing the output of an analysis like that. So for example, let's look at the first red arrow. The FGF signaling pathway has 120 genes in it. 17 appeared in our list of 500, whereas we would only have expected to see 3 by chance. So that's a significant overrepresentation of genes, and you get a tiny p-value, although you do have to recognize you're doing many tests here, so you need to do some sort of correction. And so that's the far right column. But nonetheless, you're seeing lots of significant pathways appearing in this particular analysis. So there's a couple of complicating factors here, and the first one is indicated or suggested by the second red arrow, which looks at another pathway called the EGF receptor signaling pathway. And as you might guess from the name, that's not entirely dissimilar to the FGF signaling pathway. I know nothing about either of these things, but smart people like Huaiyu know exactly what these pathways are, and they've drawn them for us at the bottom here. So FGF on the left, EGF on the right. And if you stare at them for a few seconds, you notice that, and because there's a big red box around it, there's a part of those pathways which is actually essentially identical. And what could easily be happening here is just that there's overrepresentation in that region indicated by the red box and not so much outside the red box. So what you're really seeing here is 1 positive result, not 2. So we'd like to respect that kind of complication. Here's probably a more fundamental issue, which is that if you look at the definition of any given pathway across different databases, ideally you would hope to find the same answer, but you actually don't. So here's an example in which we're looking at apoptosis pathways across the Panther and Reactome databases. And what you'd like to find is that a gene is listed in both, in that pathway in both databases. And those are the red spots that you can barely see behind all the blue and green ones. The actual reality is that most of the genes indicated as being in these pathways are listed in one database or the other database, but not both. So there's a real lack of consistency, and this is a representative picture of definition of pathways across databases. And of course, you'll get a very different answer about what's overrepresented depending on what's actually listed as being involved in that pathway. So we'd like to solve those 2 problems. And so again, we're giving a very sort of 2,000 foot this time altitude description. We're trying to develop pathway analyses that are agnostic to the specific definitions. In other words, to the specific database you look up the definition of a pathway in. And I won't go into the details of how we're going to do that. But then also, when we interpret the results of that, respect the fact that different pathways have common key modules. So the delivery or deliverable here will be pathway analyses that are much more robust and much more interpretable than existing methods. And there are a number of existing methods out there. And then the third aim is a slight change of tack. We've all got genomes. We all know they're important. It's the personal sequence of A's, C's, G's, and T's we have that makes us the unique and wonderful individuals that we are. Or is it? Because it's also true that every cell in our body has essentially that same copy, same sequence of A's, C's, G's, and T's, yet some of those cells turn into liver, some of them turn into heart, some of them turn into brain, et cetera, et cetera. So how do they do that? It's obvious there must be something above and beyond the genome going on. Or if I was Greek, I would say epigenome, above the genome. And that's epigenomics. And a particular instance of epigenomics is methylation. So this is a little figurative explanation of what methylation is. Every time you have a C followed by a G, there is the opportunity for a methyl group to attach. I think a methyl group is CH3, but I wouldn't want to put too much money on that. But it's some sort of organic thing involving carbon and hydrogens. And you can walk along the genome essentially and annotate at each CPG site or each CPG pair whether or not there's a methyl group attached. And so the little lollipop diagram here is using yellow lollipops or ones to indicate that there is a methyl group attached, and zeros or white lollipops to indicate that there isn't. And that source of variation does actually vary across cells within a tissue, or more importantly, across tissues. We know it's important. It plays an important role in determining phenotype, for example, by influencing the amount of genes that are expressed. And so therefore, it's reasonable to think that if a gene is important, perhaps the epigenome can't vary too much, because then the expression of that gene might be varying and you wouldn't be getting the answer you need to get in that particular cell. So the motivation here, or the goal of this aim, is to look for conservation of epigenetics variation, conservation of methylation for us, to indicate which genes are important. I'll show you some preliminary data to suggest that there is conservation, that it is important, but there's a number of papers we've already published. This one's due to Emil Hitveld, who's been working on this for us, and then there's a series of papers from Kim, and Daryl Shibata's up, collaborator in pathology, who's kind enough to give us the data. So here's just a quick example of what happens if you look at conservation. So this diagram, on the x-axis, you're looking at location within gene, 0 being front end and 5,000 being back end. 3 different tissues indicated in 3 different colors. And on the y-axis, we're measuring conservation. I'm measuring it here using something called Manhattan distance. The details of that don't matter, although you may be familiar with it, but you need to know that low values of Manhattan distance correspond to high conservation. So what you see is in the early stages of genes, but not so much at the end, there's an increased amount of conservation indicating, as you might hope, that there are important things going on. So the goal then is to develop statistical methods, some simple, some horrendously complicated, but really, really interesting, to help us identify conservation, and then to use these tools to find regions, genes, pathways that are conserved in particular tumors, say, from data from, for example, Daryl Shibata, to help us understand which genes are important for which tumors, because we know it varies from tumor to tumor. And so here's my slide about how we integrate with other projects. Essentially, our posterior is their prior. It's precisely the kind of data that will be the output of the methods we're developing here, that will be the input to all of the multi-omics methods that you're going to hear about this week and next week. And in addition, the results from the methods we produce will be incorporated in tools developed in Core B, and we will be using Core C and D all the time to get some of this stuff up and running and out to the general public. I think that's all I've got. There we go. So at this point, I'm going to pass you over, I believe, to Huaiyu to talk about Core B.
Thanks. I need permission to share my.
I've made you co-host. You should be able to.
Oh, okay.
You can now.
I can do it. Hold on. Let me see. Where's my? Okay. All right. All right. So can you see my slides?
Yep.
Okay. So Core B is the functional annotation core. Our overall goal is to provide functional annotations to genetic variants and to support easy access of these annotation data to the program members in their research. So I'm leading the project, and together with Paul Thomas and Juan Pablo. And there are 2 students involved in the projects. One is Caitlin, the other is Drew. So our ultimate goal is to annotate variants, and there are many tools out there. Probably many of you know about it. But one drawback for those tools is, each tool annotates one part of the annotation data, and oftentimes when a lab they want to annotate a more comprehensive set of get a more comprehensive set of annotations, they have to install multiple tools in order to do that. But what we try to do, and also the learning curve is pretty steep, and you need a lot of computational and bioinformatics skills in order to do that. So what we try to do is develop something relatively simple, and you can access it in an easier way. So what we think is, instead of asking you to do all these setup, if we can create a relatively simple query systems that you can just do quick query. That will be easy. So with that in mind, we designed the system as this. We pre-annotate the variants that are going to be used in the research, at least for now, as a proof of concept for this program. And we use co-ret data, which is the same as the haplotype reference consortium data. It's HRC variant data. It's about 40 million variants. We identify two types of annotations. One is through this tool called WGSA, and the other is more functional annotations. So the WGSA is an integrated platform developed by a group in University of Southern Florida. So what they do is they incorporate three variant annotation tools together with a number of resources, such as annotation data for allele frequency and sequence feature, and also disease and epigenomic data. So all together, we can just run this program and annotate all these variants. The other is, one thing is lacking in this platform is that most of those annotations are what is called gene model or sequence feature annotations. It will tell you where the intron exon is, where's the intergenic region, the promoter and enhancer. But what it lacks is the functional data. What I mean is, for example, what this gene is involved in, what pathway they are involved in. So we actually, my group is working closely with functional annotation or data science consortiums in the world. For example, Gene Ontology and Panther and Reactome, those are very well-respected functional annotation consortiums. I'm part of it. So we take advantage of our relationship, and then we port these annotations to our pipeline also. In addition, this core also developed a enhancer to GeneLink database that links those enhancers. Those are in the non-coding region of the genome to the genes, so that we can provide more functional annotations to the non-coding regions. And this database is called Peregrine. Let me see if I can remember what it stands for. It's predicted by experimental results, enhancer gene relationship, illustrated by a nexus of evidence. So that's how it's meant for. So we build this pipeline, we annotate this entire variant set, and we build this annotation database, 40 million about. And then we have over 600 annotation types altogether, all these annotations. So this annotation database serves as the backbone of our entire platform. And this platform, basically we have an API to access the database, and there are various different ways to, you can access these databases for the annotation. So there are few features of this entire setup. One is the accessibility. It's, there are multiple ways to access the data. There's an interactive way. You can have an interactive website to annotate the data, a small, for example, if you have 10,000 or less variants, you can just use this interactive platform, interactive website to annotate it. But also, if you are you're running R script, and you can use an R package and embed it into your script, so that when you run the analysis, you can access to the annotation data on the fly. And the let me see. The second is the scalability. So as mentioned, we right now only include one type of one data set, one variant data set. That's the HRC. But the way the database is set up, it's in an Elasticsearch framework, we can actually technically we can increase the data size tremendously. For example, we can include the entire DB SNP. We can still give very high performance on the query and search, and basically we can scale up tremendously. The third is flexibility. That means you can access and annotate the data based on what you need. You can mix match annotations from different resources. That's something that if you have other tools, usually you cannot do that. But here, you basically can use the interactive web interface to go through these 600 or so annotation types, and you find the ones that you like, and you can generate a configuration file and use that. In the future, you can annotate just based on whatever the annotation you choose. So that's the flexibility part. And so the annotation platform is called an Annotation Query, or ANOQ. It can be accessed through this website, anoq.org. And there are links to the interactive website. You can go into the API to find out scripts to do query, and also there are packages. There's a detailed seminar we give during the summer. It's recorded and available on YouTube. If you want to know how to use this platform, I encourage you to go to this. It's in the Biostats seminar YouTube channel, and you can go in there and find this presentation and go through the details. And then the Peregrine project I mentioned to you, that's an Enhancer to Gene project. Sorry, I just want to mention, this project was done by Zhu mainly. It was a tremendous amount of work to put all these together, and he's the main developer doing this, and with some help from other engineers to set up the website, etc. And Peregrine project is done by Caitlin. It's as I mentioned, it's an Enhancer to Gene relationship database. The data is available. You can browse and search, and actually a lot of the data is available on the Panther website. You can use Panther to search all these different enhancers, and then you just need to provide a VCF file. You can also download the entire data and to your computer, and you can analyze or use it programmatically. And there is also another seminar presented by Caitlin. It's also in that channel. I encourage you to go there and take a look for more details. So in this renewal, what are we going to do? Basically, we have three aims also. The first is our focus is mainly three things. One is the quality, one is the performance. One is accuracy, actually, the data, and the second is performance, and the third is support. So the accuracy is mainly on the annotation data, right? We want to keep all the data up to date. We want to improve the quality of the Peregrine database. And we also want to add additional annotations and add additional variants. But one thing, this is something we added recently, is we want to provide also data validation, data control, quality control, and evaluation, mainly to develop some kind of benchmark studies so that we can give user more educated information that they can make decisions what kind of annotation they want. Because the database is just so big, how this is something some of the users come back and ask us, with that, how do I choose it? So I think this is the last part is going to be very useful. And the second is with the increase of the annotation type and variants, the database will increase also tremendously. As I mentioned, we use the Elasticsearch framework, so the performance should be very good and we can scale it up very well. But at the same time, we also want to ensure, the performance is good. And we want to, and when there are many, many users, we also want to publish it and make it available to the general public. So when we increase users, one thing is the performance will be impacted. So what we want to do is several things to accommodate that. So one is we want to develop software packages so that people can know exactly what they need to access and use those packages and to access data directly. That will increase, alleviate the performance issue a little bit. And the other is we really want to make sure that the performance is good by improving the hardware and also the Elasticsearch capability. And the last is the security with a lot of users. And one issue is how we can ensure the IT security. And so we are going to beef up that. And the third aim is really about training and outreach. Not only we are going to outreach to our users, but also we are going to reach out to the sources, like people who provide data, the tools that we use in there. We would like to send feedback to them and ask them to get involved also. So this is where we are in Corby, where we stand in this overall picture in the renewal. We are going to provide annotation data in the very beginning, but also Core B will work together with Core C and D to help analyze the data. That's pretty much it.
Well, thank you, Paul, Jim, Huaiyu. Got a couple of minutes left in this block. I'm certainly willing to hang in there a little bit longer if there are additional questions. Floor is open. And as I said earlier, next week, come back to hear about projects 1 and 2 and their supporting cores. We've been recording this talk, as we will do for all of them, and if you missed anything, they will be posted before long on, this recording will be posted on the YouTube, the Biostat Division YouTube channel. Well,
so I do want to mention one thing. So Yu, who is the student working on the anal cube, and he's taking a leave. So I am looking for student to work on that project. Just want to.
I think the same could probably be said about several of the other projects as well. So volunteer.
Yeah. If you want to know more, I'll be happy to talk to any student who are interested.
Likewise.
We're not hearing any other questions. I am going to stop recording at this point, but leave the thing open so anybody can say whatever rude things they want.